So today we are going to study about the RNA, that is ribonucleic acid. It is single strand and it is prepared over DNA at the process of the transcription. So now we are going to study how many types of RNAs are there. Now see, there are 6 types of RNA. Messenger RNA. Okay, this RNA carries the genetic information which is coded from the DNA to outside for the translation process. So messenger RNA contains codons, means they contain the sequence of the nitrogenous base which will be translated by the process of the translation. Ribosomal RNA. These RNA, they are the structural component of the ribosomes, because as we know, ribosomes are made up of RNA and protein. So they are made up of ribosomal RNA. Then third one is transfer RNA. Transfer RNA is also known as adapter molecule. Okay, it is also known as adapter RNA. It is also known as soluble RNA. Please remember the short form what is used here. This small s is very important. It is soluble RNA. Then for the splicing, you want snRNA, small nuclear RNA protein. Small nuclear RNA. Then there is small cytoplasmic RNA also. And next is your genetic RNA, which is seen in the viruses. Because generally the viruses are the one which are having the genetic material as RNA. So you have to remember here, see the single S means soluble RNA. If SN word, small SN comes, it means it is small nuclear RNA. And if a small SC comes, that means small cytoplasmic RNA. When they are giving in the option, then you have to see it very carefully. Now see, out of these 6 types of RNA, these 3, they are very important. Okay, these are the major types of RNA. One is a messenger RNA, okay, ribosomal RNA, transfer RNA. They are very, very essential. Now see, ribosomes are made up of ribosomal RNA and protein. And you know very well, there are 2 types of RNA. 70S ribosome is seen in prokaryotes and is seen in mitochondria and chloroplast of eukaryote. So it is present in the mitochondria and chloroplast of eukaryote. And 80S is present in the cytoplasm of eukaryote. '70S is made up of 50S and 30S. 80S is made up of 60S and 40S. So now we have to study how many types of ribosomal RNA are there. Okay, see the board carefully. Here you will find 23S ribosomal RNA. Here you will find 5S ribosomal RNA. Here 16S ribosomal RNA. Here you will find 28S ribosomal RNA. 5S ribosomal RNA. 5.8s ribosomal RNA. Here you will find 18s ribosomal RNA. Now you have to remember here, see carefully, you have to remember this is working as ribozyme, means it will help in the peptide bond formation in case of prokaryote. And here this will work as a ribozyme. Why I'm using word ribozyme? Because these are the RNAs which are working as an enzyme. So we call them as peptidyl transferase also. Okay, so here the RNA is working as an enzyme. Which enzyme? Peptidyl transferase enzyme. So we call them as ribozyme also. So these are the types of RNAs which are present in the ribosomes. Now see the structure of messenger RNA. If you see the messenger RNA, we have already discussed the fifth end will be there, which is identified by the capping. Okay, this is your fifth end. And the third end will be identified by the poly A tail. And in between that, if you see now, see carefully, this area will be the one which will be translated. Means this will be the language will be converted into the sequence of amino acid. And from here they will start. So this is known as start codon. And this is known as stop codon, from where they will stop. But this part, check the board carefully, this part will not be translated. You will not do the translation of this part, neither we will do the translation of this part. So this part are known as UTR. They will not be translated. Okay, so these are UTR. Now what does UTR stands for? They are untranslated region. So from the diagram it is clear that UTR is present in front of the start codon. Means they are present at the fifth end upstream of start codon. Okay, and here you can say that they are present at the back of stop codon at the third end. So UTR are present before the start codon and they are present at the back of the stop codon. And this area, this is completely the translation unit. So this is your translation unit. So this is the one which is totally translation is done for this area. So we will say translation unit is flanked by start codon and stop codon. Okay, so this is the messenger RNA which is going to be translated and a sequence of amino acid will be arranged with the help of transfer RNA. So messenger RNA has got capping. Okay, the capping is done. Then tailing is done. Capping is at the fifth end. Tailing is at the third end. And only this area will be translated. See the board area? Only this area will be translated from here till here. Okay, stop codon is for the termination of the translation process. So these are not doing any work, they are just giving us idea. Now you have to stop the translation. So we will say translation unit is flanked by what? It is flanked by start codon and is flanked by stop codon. I hope this structure is clear of messenger RNA. Now see the transfer RNA. If you see the diagram of transfer RNA, this is actually 2D structure of transfer RNA, okay? So this 2D structure of transfer RNA is clover leaf-like, okay? So 2D structure is clover leaf-like model. Now you have to see the board very carefully. If you see here at the fifth end, you will find G is present. The fifth end is starting with the purine. Okay, fifth end G is present, which is starting with the purine. And at the third end, see carefully, at the third end, These are the three nitrogenous bases which are single strand. And sequence we have to learn CCA, because third OH. Here only the amino acid will be attached. At the third OH amino acid attachment will be there. So we call this site as amino acid attachment site. So this sequence is very, very important. Please learn it carefully. At the third and which base is present? CCA. We will not say ACC. Sorry, we will not say ACC. Is it clear what we have to speak? We have to speak CCA. That we have to speak, and amino acid is attached here. So if you see carefully the diagram, at the fifth end and at third end, both end, purine is present. Okay, now this is the site where amino acid will be attached. Now this is your loop areas, and this is the one loop area where anticodons are present. Anticodons, they are the area which are complementary to the messenger RNA, which was bringing the codon. Because messenger RNA, you know very well, the messenger RNA is bringing the sequence of nitrogenous base. And sequence of nitrogenous bases, 3 sequences of nitrogenous bases means 1 codon. So here, to read them, it is anticodons are present on transfer RNA. So I hope this topic is clear. If they ask the question, where is anticodon present? So your answer will go, they are present on the transfer RNA. And where is codon present? Codons are present on messenger RNA. Is it clear? See this structure. This structure of transfer RNA is 3D structure, okay? And this is a 3D structure which is giving a inverted L. This is inverted L shape. And this is your transfer RNA with anticodons. These are the anticodons, and this is the amino acid attachment site. So such type of diagram will be drawn where we'll be explaining you the translation. I hope this topic is clear to you. All the 3 types of RNAs, detailed structure. Thanks for watching class. Still, if you have any doubt, then you can message me. I'll try to help you out. If you liked it, please give it a thumbs up and subscribe to my YouTube channel.